All right, so let's continue with gluconeogenesis, which is not exactly the reverse of glycolysis, but it's a way of turning pyruvates back into glucose. We can't obviously use the 10 steps of glycolysis because, as we said last time, the 3 of them are irreversible. So gluconeogenesis is in fact using the other 7 steps exactly. We need to find a way around the 3 irreversible steps. So if you learn glycolysis first and know those 10 steps, gluconeogenesis isn't that difficult. It's just learning the roundabout way of getting by those 3 irreversible steps. Okay? So what we're going to do is make glucose from non-carbohydrate or non-sugar precursors. So pyruvate is not a sugar, so we're going to make glucose from pyruvate. Now we could make glucose from, say, maltose, but that is not gluconeogenesis. That's just glucogenesis, right? We could make glucose from starch. Again, not gluconeogenesis, because it's already a complex sugar. It's a sugar already. We're just breaking it up into its sugar parts. Here we're making a sugar anew. That's what the neo means in it. So we're making sugars from non-sugar starting materials, okay, such as pyruvate. So the overall reaction at the bottom here is we're going to take 2 pyruvates, that's all 6 carbons, and make a glucose. Along the way, we're going to use 7 of the 10 reactions that we just studied with glycolysis, all the reversible ones, including step 6, where we do the redox reaction. So we're going to use 2 NADHs converted back to NAD+, and that's because we need to do that for 2 molecules at that stage. But we're also going to have to convert 4 ATPs and 2 GTPs into their hydrolyzed products. So in glycolysis, we had a net gain of 2 ATP, where here we're going to have a net cost of 6 ATP equivalents. So this is expensive. It costs 3 times as much to make a glucose as you're going to get out of it in glycolysis. So why would you want to do such a thing? Well, maybe we need glucose for something else. Maybe I don't need glucose right now, or I don't need ATP right now. I have an abundance of ATP right now. So if I have all this pyruvate, let's do something with it. I can turn it into glucose and store it for later, because I don't need to turn it into acetyl-CoA right now. And that's irreversible. Or I could take the glucose and make other things with it, like maybe some ribose. I could make ribose from glucose. Maybe I need to make some more nucleotides. Or I could make other things from glucose. There's lots of things you can make from the metabolites in glycolysis. Remember back to our metabolism branch chart, right? There's lots of lines coming off of those molecules from glycolysis. Let's use them to make the other things we need, right? So here's a way of storing pyruvate as glucose. Yes, it's costly, but I'm not worried about fuel right now. I'm abundant in fuel. I want to store it for later. I don't want to waste it. Okay. So our first step is, where can we get a non-carbohydrate precursor? Some examples. Well, one would be lactate, right, at the very top here. So we have lactate in our skeletal muscle. We send it to the liver. We can convert it back to pyruvate in the liver. That's not a problem. And then we can make our glucose from pyruvate from there. So lactate's also another potential source for this. Some amino acids can be used for this. In fact, all but 2 of them could be used to make more glucose, or 2 that cannot. We'll learn those later. And then our triacylglycerols, at least the glycerol part, right? Not the fatty acid part, because we know that can't be turned into glucose, but the glycerol part can, right? So we take our glycerol, and it's just 2 steps away from being dihydroxyacetone phosphate. So our glycerol, if we phosphorylate it using an ATP, now we have glycerol phosphate. And then if we oxidize that middle carbon from a secondary alcohol to a ketone, we now have dihydroxyacetone phosphate. And if I do that for another glycerol molecule, make a second dihydroxyacetone phosphate, I can convert one of the 2 into the aldehyde form, and then together those could form, through aldolase, my fructose 1,6-bisphosphate. So I can send it right up the tree of glycolysis from glycerol. Fatty acid parts we can't turn into sugar, but some amino acids we can, in fact all but 2 of them, and lactate we can, and pyruvate we can. Okay. So where does this take place? Well, it happens in many cells, but mostly in the liver and kidney. We'll focus mainly on the liver. Brain, skeletal muscle, heart muscle, red blood cells can do all this too, but mainly in the liver. Right? So the purpose of the liver is to serve all the other organs, right, and to filter things for the body. So we're worried about the liver here. Okay? It is not simply the reverse of glycolysis. Don't think it is. And the reason it can't be is because we have 3 irreversible reactions that we can't go backwards in. So right now with steps 1, 3, and 10 of glycolysis, hexokinase, PFK1, and pyruvate kinase. And what happens in each of those steps to make them reversible? Well, in number 1, we had hydrolysis of ATP, which was a very energetic thing, negative 7.3 kilocalories per mole, right? Same thing in PFK1. And in step 10, we got rid of a very unstable phosphoenol pyruvate, which had more energy than ATP. So in each of those cases, to drive them backwards would be to either, in steps 1 and 3, assemble an ATP with not enough energy to do it, or in step 10, use an ATP energy, 7.3 kilocalories per mole, to put a phosphate back on pyruvate and make phosphoenol pyruvate, which is not going to happen. It takes more energy than that. So in all 3, we're lacking the energy to go backwards. So let's bypass them. Okay. So here's our glycolysis and gluconeogenesis one more time. So glycolysis, this time is going to be on the right-hand side in this figure, right? So it's red still, but it's on the right-hand side. And it shows all 10 steps once again, and the 3 arrows indicate the 3 irreversible steps. Okay, these are the ones we need to bypass. All the others you see by the red and blue arrows are completely reversible, right? You can go backwards and forwards through all of the other reactions, including step 7 and step 6, right? So everything except 1, 3, and 10 are free to go in reverse. So we can really ignore those for this part. How do we get past steps 1, 3, and 10? Well, let me tell you a little bit about how I'm going to number these for you so we don't get confused. So by convention, we're going to go from top to bottom in glycolysis, step 1, 2, 3, all the way to 10, right? Listed top to bottom. That's what we just did in the previous lecture. For gluconeogenesis, so as to not be confusing, when I say step 6 in gluconeogenesis, I mean the same step 6 that's in glycolysis. So we all agree that step 6 is that same reaction. So therefore, the reactions in gluconeogenesis count down instead of up, right? So the last step in gluconeogenesis is not step 11 of it, it's actually step 1. So what I'm referring to here is, let's pick step 2 for instance. So step 2 is reversible. It turns glucose 6-phosphate into fructose 6-phosphate. That is step 2 of glycolysis right here. It's phosphoglucose isomerase. But it's also phosphoglucose isomerase for gluconeogenesis, step 2 I'm calling it. So if you read these 1 through 10, top to bottom of glycolysis on the right, I'm going to read these 10 through 1 on the left in the process of gluconeogenesis, so that I'm not confused when you say, well, what about step 8? Well, I know what you mean. Step 8, exact same one I'm talking about, right here, step 8. Right? It's step 8 in both directions, because we count up in glycolysis and we count down in gluconeogenesis. The problem with that is it's backwards, but the real problem is there's actually 11 steps in gluconeogenesis. To bypass step 10 of glycolysis requires 2 reactions in gluconeogenesis. And in fact, it's more than that when we get into it. So we'll call these step 10A and 10B if you want, right? And then we do 9, 8, 7, 6, 5, 4, 3, 2, 1. Okay, so going top to bottom is glycolysis on the right. From bottom to top is gluconeogenesis on the left. We're going to count the steps down so that the common steps get the same numbers. Okay, so how do we bypass these? Well, some of them are very simple. Bypassing steps 1 and 3 are trivial. They're very, very easy. So how do we bypass step 1? We simply just cut off the phosphate. Now, I don't want to be doing glycolysis and gluconeogenesis as step 1 at the same time. Hexokinase putting the phosphate on, and glucose 6-phosphatase, the enzyme, cutting it off continuously, because I just continuously waste ATP. That's not a good idea. So we don't actually go backwards and make an ATP in gluconeogenesis, but we just cut off the phosphate. The same can be said for step 3. PFK1 puts on the phosphate, and then fructose 1,6-bisphosphatase cuts it off. And that's what phosphatases do. They just cut off phosphates. So the reversal of, not reversal, but the way around steps 1 and 3's irreversibility is simply to cut off the phosphates. Those are trivial answers, right? You just cut off the phosphate. The real meat of gluconeogenesis is bypassing step 10. How do we go from pyruvate at the bottom back to phosphoenolpyruvate, that very unstable molecule? That's not easy to do at all. We see that's where we're going to invest almost all of our energy, is making this molecule. But then it can immediately go up the tree using step 9 to make 2-phosphoglycerate and step 8 to make 3-phosphoglycerate, all running in reverse. And if we get back up to fructose 1,6-bisphosphate here, we can just cut off the phosphate, isomerize it to glucose, and cut off the phosphate again. That's trivial to do. Okay? So we'll focus most of our time on how to bypass step 10 at the bottom, going from pyruvate back to phosphoenolpyruvate. Okay? So at the bottom here, I have the molecule pyruvate drawn for you again. So this is showing the gluconeogenesis side of the tree. And I want to get back up to phosphoenolpyruvate. So the way I do that is I need to first turn pyruvate into oxaloacetate. Right? So that's this first step going up. Pyruvate has 3 carbons. Oxaloacetate has 4. So I've clearly added a carbon. And I did so at the expense of an ATP molecule, because putting this other carbon, this other CO2 or bicarbonate on was not easy. Right? I had to deprotonate a methyl group and put the carboxyl group on in its place. Right? So putting this on required the expense of an ATP. Right? No phosphates were added to the molecule net, but a carbon was. And then in the next step, we're going to lose that very same carbon I just put on. You're going to see this is a common theme in biochemistry. Put something on just to lose it. Right? Putting this carboxyl group on was not favorable, and it's very favorable for it to leave because it's beta keto to the carbonyl, whereas the one that was already there is still alpha keto. Okay, so I'm willing to lose the CO2 now because it's beta keto, and when it loses this, the electrons rearrange and formed a carbon-carbon double bond, making this O into an O-, which will be easily taken up by a phosphate. Okay, but, well, I say easily. It's not so easy. It's going to require the expense of another GTP to donate that phosphate. Okay, in the enzyme that does this, the phosphoenolpyruvate carboxykinase, that's a mouthful. It's abbreviated PEPCK or PEPCK for phosphoenolpyruvate carboxykinase, is once again named for the reaction in which we consume the GTP. Okay. Right, and then lastly at the bottom, where I have pyruvate and oxaloacetate, a lot of the amino acids I talked about that can be introduced to make glucose, so lactate and some amino acids can be turned into pyruvate, those with 3 carbons, and some can be turned into oxaloacetate, those with 4 carbons, and then I can turn those into glucose by this pathway. So some amino acids, in fact almost all of them, could enter the glycolytic pathway in some form or another. Okay? But after that, once I've made my phosphoenolpyruvate, it's very easy. All these are the reverse of the enzymes of glycolysis going up the tree. Okay? And at the top of the tree, when I finally get to fructose 1,6-bisphosphate, it's very simple to make fructose 6-phosphate. Just hack off the phosphate. Right? So I use a water molecule and an enzyme, and I chop off the phosphate, and I'm at fructose 6-phosphatase, or fructose 6-phosphate. Then I isomerize that in step 2, the reverse of step 2 of glycolysis, which is totally reversible, to glucose 6-phosphate, and again use an enzyme and a water molecule to chop off a phosphate. So like we did with glycolysis, let's decide what kind of what class of enzymes are these things. Right? So let's do these two here. Pyruvate carboxylase would be turning pyruvate into oxaloacetate, and making a new carbon-carbon bond. What class would this be?
Ligase?
It's a ligase, right. We haven't seen a ligase yet. So this is the first ligase we've seen. We didn't have any of these in glycolysis. Right? And then PEPCK, or phosphoenolpyruvate carboxykinase, would be a what?
Transferase.
Transferase, because that is doing other things, I agree, but it's also transferring a phosphate. So it's going to get a transferase nominator. So we're going to put a phosphate on, so we're going to call it a transferase. Right? And then these two are something we haven't seen yet in glycolysis. We have seen them before, but we haven't seen them in metabolism yet, except for the first lecture we did. But we're just cutting off a phosphate using a small molecule. What kind of enzyme is this? What class?
Hydrolase.
Hydrolase. And the same thing up here, another hydrolase, right? Not because they use water. So don't think they're hydrolases because they use water. That is not true. They're hydrolases because they use a small molecule to break a single bond. The most common small molecule to use is water, of course, but it's not the only one. We'll see some others later. So just because you see water, don't think hydrolase, because if you go back and look at enolase, run in reverse, right here, it is not a hydrolase. This is still a lyase. So don't think water means hydrolase. Please do not associate those two directly. Okay? And here's where our glycerol would have entered the pathway had we oxidized it and phosphorylated it, and we get dihydroxyacetone phosphate. Okay? So let's go to the first enzyme in this and do it in detail. So pyruvate carboxylase, this is the very first step in gluconeogenesis. We could call it 10A or 10B if you want to call it that. But it takes pyruvate and makes oxaloacetate. Okay? This is a metabolically irreversible reaction. Again, I know the arrow looks like it goes in both directions, but in the cell, it only goes in one direction. Okay? So it takes pyruvate and turns it into oxaloacetate. In a dying cell, it will go in the other direction. But we're talking about a healthy cell. So it's metabolically irreversible in the normal condition. Right? The problem here is this takes place in mitochondria. Right? Where did all of glycolysis take place?
Cytoplasm.
Cytoplasm. Is there any barrier from the cytoplasm to being in the matrix of the mitochondria? Or can I just go from one place to the other rather easily? If you were a pyruvate molecule, is there any barrier to you?
The membrane?
Yeah, there's in fact two membranes, but most importantly the inner mitochondrial membrane. Right? The outer one has pores in it, it's not a problem. But the inner one is watertight. Right? So the pyruvate molecule can't cross that membrane on its own. So first thing we need to do is get the pyruvate in the mitochondria. So we need to have a channel, some kind of transporter that gets pyruvate from outside to inside. Okay? So we'll use one of those. It doesn't cost us anything, but we have to have a channel to get pyruvate inside. Once inside, that's where we find pyruvate carboxylase, which uses pyruvate and a CO2 in the form of bicarbonate, right, and an ATP molecule in water to make oxaloacetate on the right. Right? Is oxaloacetate a stable molecule? Look at its structure and tell me, do you think, is oxaloacetate on the right stable?
No, because of the two negative charges.
Well, two negative charges doesn't make it completely unstable. In fact, they're as far apart as they can get from one another.
Also resonant and stabilized, aren't they?
So what would make it unstable?
Beta keto arrangement of the carboxyl and the carboxyl and the third carbon.
The fourth carbon.
Third carbon to first carbon.
So the carboxyl at the bottom here, relative to this carbonyl, is beta keto. Right, so there's a double bond here, single, single, and then a double bond. Right, so these are beta keto to one another. And the top one is alpha keto to it, just like it was back over here, between carbons 1 and 2. Carbon 1 and 2. But carbon 4 down here, the one we just put on, is beta keto to the carbonyl. Right? Or you can say the carbonyl is beta keto to it. Relatively, they're beta to each other. Right? So this is not stable. Right? So we need to do something with this. Well, we're in the mitochondria right now. The enzyme that does something with it, the PEPCK, is located back in the cytoplasm. So I need to get my oxaloacetate out of the mitochondria. We only came in here to get carboxylated. We need to get back out into the cytoplasm. But do you think oxaloacetate can cross the membrane?
No.
No, there's no chance it's going to cross the membrane. It's even worse than pyruvate, right? It's got one extra carbon and it's got another negative charge. So what we need to do is convert it to something that can have a shuttle across the membrane. So we're going to convert oxaloacetate into malate. We're going to reduce it once. We're going to reduce this, I'll show you a picture of it in a second. We're going to reduce this carbonyl to a secondary alcohol. right? So we reduce oxaloacetate to malate, and then we export the malate, which we have a channel for that. And then once it's outside in the cytoplasm, we oxidize the malate right back to oxaloacetate. So it was just a trick to get it out of the mitochondria. So once it's out, and oxaloacetate again is reformed, oxidized here, we're going to use PFKI to do two things to it. The very CO2 we just put on is going to leave, right? So the CO2 leaves, and the bond, not the lone pair, but the pair of electrons between those carbons migrates to the other pair of carbons, between numbers 2 and 3 now, which means you can't have a carbonyl anymore. So that carbonyl becomes an O-, and then we use a phosphate from GTP to put a phosphate on that O-. Okay? The O- actually attacks the phosphate. Right? And that's what PFKI does. So we lose the CO2, that's a favorable thing. We hydrolyze a GTP, that's a favorable thing. And we make a very unfavorable or unstable phosphoenolpyruvate. Overall, the delta G of this is still not negative. It's crazy. We're losing a CO2, we're hydrolyzing a GTP. You know how that's a negative 7.3 right there. And the overall delta G of this is still ever so slightly positive. So this is not an easy reaction at all. And in fact, remember we had to spend an ATP back here to put the CO2 on just so we could lose it later. Right? That's how energetically unfavorable this molecule is. Right? Is this reaction reversible? No. Right? So again, this is going to be irreversible. Okay. So here's how the first reaction works, pyruvate carboxylase. We need to get a CO2 attached to what currently is a methyl group. Right? So we need to deprotonate the methyl group, which is not an easy accomplishment. And we need to activate our CO2, because CO2 is rather inert. Right? It's a nonpolar molecule. We need to bend it so we have access to its carbon. Right? So we turn it into a CO2 attached to a nitrogen. So this is a CO2 up here, right? That's been attached to a nitrogen. It's now a carboxyl group, right? So how do we do that? What nitrogen should I use? I don't want this to be a permanent attachment, just temporary, so I can bend it into this form. Instead of being linear, like CO2 is, it's now trigonal planar, so I can attack it. So we use a cofactor for this called biotin, right? And biotin has a couple parts. Biotin has a really long tail, as you can see over here on the right. A lysine amino acid R group is sticking out. There's its 4 carbons and its amino group. And then there's 1, 2, 3, 4, 5 carbons, 3, 4, 5 of its tail here. So it's got a 5-carbon valeric acid tail. Valeric acid just means 5-carbon carboxylic acid tail attached through here. And you notice this is going to be a carbonyl. So this is our like an amide bond attachment. It's not a peptide bond. It looks like one, but it's not. Right? Here's our 5 carbons again. And then we have a pair of fused rings. You can see it on the right here as well. There's 2 5-membered rings that are sharing a side. Right? So the one at the top over here and the bottom over here, or side over here, is 4 carbons and a sulfur. That's called a thiophene ring. Right? There is a double bond in it. So it's got a sulfur, there's a thio part, phene indicating it's got a double bond, so it's a thiophene ring. The other 5-membered ring is made up of 3 carbons and 2 nitrogens, again with double bonds in it, and it's called a urido ring because it resembles urea. Right? The middle part of it looks almost like urea. We have a carbonyl and 2 nitrogens. So a thiophene ring and a urido ring. On the urido ring, this nitrogen is where we're going to attach our CO2. So the purpose of biotin here, it's a cofactor, coenzyme, of this enzyme, pyruvate carboxylase, and its job is to carry the CO2 for me. Right? So we said back on a chart, if you look on other lectures, that one of the roles of biotin as a cofactor is to carry CO2, carry 1 carbon units in the form of CO2. That's what it's doing for me. Okay? So how does this thing work? We said we need to deprotonate a methyl group. That's not easy. Right? So I need some base, and we can't just grab a base off the shelf. This has to be done in a cell, so we can't have something crazy. Right? So we need some base as part of the amino acids to remove the proton from this, making it a very energetic CH2 with a minus charge, which will immediately collapse, right? Or immediately attack a CO2. Right? So we have a carbonyl, or sorry, a CO2 nearby, which has a carbonyl in it. And if I deprotonate this methyl group, the CH2 with a negative charge will immediately attack that carbonyl, forming this new carbon-carbon bond. And when it does so, the CO2 releases its bond from the nitrogen, and that nitrogen can be used to grab another CO2. Okay, so that's how we get this on there. The hard part is to remove this methyl group, and that's where the expense of the ATP comes in, to remove the methyl group at the same time as activate the CO2. I put a phosphate on the CO2 to make carboxyphosphate, which is not stable, and biotin's nitrogen attacks the very same carbonyl. This carbonyl is getting tired of being kicked in the back, but that's all we do to carbonyls, and it releases the phosphate. Okay. So all of this took place in the matrix of the mitochondria. So I said we had to get our pyruvate in there from the first place. So crossing the outer membrane is not a problem. Crossing the inner membrane, we had to have a channel for that. So we got pyruvate in. We'll talk about channels, which ones let it in later. We carboxylate it, right, to make oxaloacetate. We can't get oxaloacetate out, so we convert oxaloacetate to malate. This is actually one of the steps in the citric acid cycle we'll talk about later, run in reverse. We're repurposing it. So we turn oxaloacetate into malate. It's just a reduction of this ketone to a secondary alcohol at the expense of an NADH, right? So NADH donating the electrons to make this into an alcohol. The malate can then get out. We'll cover that in our shuttles again later. The malate outside converts right back into oxaloacetate, again at the expense of an NAD plus getting reduced to NADH. So effectively, I've turned one NADH into NAD plus and one NAD plus into NADH, but in different compartments. So they're not in the same compartment. This is called the malate shuttle, where I get pyruvate to oxaloacetate. In order to get that oxaloacetate out, I first convert it to something I can transport, malate, transport it, and then oxidize it back to oxaloacetate. Don't forget this step. It's not listed in the overall reaction because we didn't really do anything. We just moved it from one space to another. But we did our conversions, our redox reactions in different compartments. So the NADs and NADHs aren't necessarily equivalent. They're in different compartments. So don't forget this conversion step, this malate shuttle. Okay, once outside, we have our very unstable oxaloacetate once again, and we need to do a reaction that turns it into phosphoenol pyruvate. Okay, so to do that, we use PepCK one more time. Right, we lose this CO2 at the bottom. It decarboxylates because it's beta keto. The electrons in that carbon-carbon bond migrate to the other carbon-carbon bond above it, forcing this carbonyl to become O-. Right, this O- then attacks the phosphate on GTP, leaving a GDP behind. So that's how we make our phosphoenol pyruvate. The byproducts of that are again GDP left over from the hydrolysis, or not the hydrolysis, but the losing of one phosphate, and the decarboxylation of the beta keto group to make CO2. Okay, so same reaction shown here. How does this work? We do them simultaneously. We lose the CO2 and we phosphorylate it in the same step. Those two reactions are now coupled. Putting this phosphate on is not favorable. There's another negative charge nearby, and it's in a planar arrangement. It doesn't want to be here at all, and it's putting on a phosphate in a beta keto arrangement. Not favorable. So at the same time, I need another reaction that is very favorable, like the loss of CO2 from a beta keto. We're trading one beta keto for another. But losing this CO2 is favorable because it's a molecule that can fly away. Its entropy is increased. So doing all of that, we still get a delta G of plus 0.8. Right? Now, if I wanted to try to reverse pyruvate kinase directly, so taking pyruvate and a phosphate from ATP and making phosphoenol pyruvate, the delta G would be plus 31 kilocalories per mole, which is never going to happen. Right? But a plus 0.8 kilojoules per mole, or 31 kilojoules per mole, plus 0.8 kilojoules per mole is reasonable, right? That can happen. I can get over that plus 8 just by Le Chatelier's principle. These are small, near zero numbers. But plus 31, I'm never going to go backwards. Okay, so I'm using the decarboxylation here to drive what would be otherwise a very unfavorable reaction, right? Because CO2 is an excellent leaving group, especially when it's beta keto. Okay, so looking at these one more time, we've covered the reaction at the bottom, which is the bulk of this lecture, turning pyruvate into phosphoenolpyruvate. Don't forget that you had to go into the mitochondria to turn into oxaloacetate, take the malate shuttle to get out, return to yourself to oxaloacetate, then do the PEP-CK reaction, which again is still not very favorable. Right? All that combined just to bypass this very unfavorable and irreversible pyruvate kinase step. Okay? Let's move on to two easier steps. Let's bypass steps 3 and 1 up here. Right? So those are very, very simple. We're going to take a fructose 1,6-bisphosphate, add a small molecule, in this case water, and just lop off the phosphate. Done. Simple hydrolysis. So this is our hydrolase. This should be reminiscent of our proteases, which were also hydrolases. But this is the first one we've seen in metabolism other than our digestive enzymes. So simple, cut off the phosphate. Okay? And now fructose 6-phosphate can easily be converted to glucose 6-phosphate. And that's typically where gluconeogenesis ends. We don't cut off the last phosphate. So going back, we're talking about this reaction. We do this, but then this is where we stop. We hardly ever do this first reversal. We don't use glucose 6-phosphatase at all. There's only one cell type that will want to do that. Remember, as soon as glucose gets in the cell, we phosphorylate it. If this enzyme is around, it's going to dephosphorylate it, which we will then rephosphorylate, and we're wasting ATP. So glucose 6-phosphatase is generally not made in most cells because we don't want to lose our glucose. In liver cells and in kidney cells occasionally, because the kidney originally did this before the liver developed. But let's focus on liver. Liver cells sometimes want to export glucose, and to do so they can't have it as glucose 6-phosphate, so they need to cut off that phosphate. So only liver cells, for the most part, some kidney cells do too, but mainly liver cells express this enzyme, glucose 6-phosphatase. So the only time you'd ever want to do that is if you want to export it, and that's done by our liver, which has to maintain our blood glucose concentration. Okay? What normally happens to the fructose 6-phosphate in almost all other cells is we convert it to glucose 6-phosphate, and then we store that as glycogen, which we'll cover for your next test. Right? So we'll turn our glucose 6-phosphate into glycogen molecules if we want to store it. Okay? So let's say we want to export it, however. So now we're talking about a liver cell mainly. So we want to cut off this last phosphate, and this is irreversible. You can't put it back on by the same enzyme. You'd have to spend an ATP to put it back on. So don't cut this off unless you intend to export it. So only liver cells is the only tissue that expresses this enzyme, glucose 6-phosphatase. If it were expressed in other cells by mistake, what would happen to those cells? They'd rapidly run out of ATP because they'd be fighting. Glucose 6-phosphatase would continually cut the phosphate off. Hexokinase would continually put it back on. Eventually the phosphatase wins the fight because it will run out of ATP. So we don't want to express that in a regular cell. In fact, the glucose 6-phosphatase, when it's made in liver cells, is made fused or bound to the ER. Right? It's complex to the ER. So why would you do that? When you have a glucose 6-phosphate that you dephosphorylate, you cut it off and make a glucose, the only point to do that is to export it. So if you release it into the cytoplasm, it's going to get phosphorylated again at the waste of an ATP. So let's cut off this phosphate and immediately package the glucose for export. So I need to do that near the membrane, near the ER, so I can export it, and the transporter that exports it. Okay, so let's look at the overall reaction. We start with 2 pyruvates, and if we want to turn those directly into glucose by glycolysis, so from bottom to top of glycolysis would be plus 75 kilojoules per mole, which is never going to happen. But if we do it by gluconeogenesis, it has a negative 38 kilojoules per mole. Why is there such a huge difference in these two? It's like 100 kilojoules per mole difference. It's because we're hydrolyzing 6 ATP equivalents here. Right? Here we get 2 out of it. Here we have to input 6. 4 ATPs and 2 GTPs. Now let's look, where did we do those? Go back to a chart of all of it. Where did I put those in? Well, I don't put anything in here. I'm just cutting off phosphate, so it can't be those steps. It must be down here at the bottom. So turning pyruvate into oxaloacetate costs me an ATP. But remember, I'm doing this for 2 pyruvates. So there's 2 of our ATP right there. Then I convert oxaloacetate into phosphoenol pyruvate. Well, the carboxykinase step, right, uses yet another GTP, or in the case of bacteria, it's ATP, right? But that's a pair of those again. So this is a total of 4 ATP equivalents right here. But I said 6. Well, if you remember, step 7 of glycolysis made a pair of ATPs for the 2 times we do it. I'm also going to have to consume those to go backwards. But this one is reversible. So I spend 1 here, pyruvate to oxaloacetate. I spend 1 oxaloacetate to PEP. Then I spend 1 from 3-phosphoglycerate to 1,3-bisphosphoglycerate. And since I did this for 2 molecules of pyruvate, that's a total of 6 to get to here. Right, now cutting these off doesn't cost me any ATP, right, if I plan to export the glucose. Right, so that's where I get 6 total ATP equivalents here. Right, ATP and GTP, same energetics, so I'm calling them all ATP equivalents. So the 6 high energy phosphates had to be spent. Okay. All right, regulation. This one's very simple, just like glycolysis was. Remind me on glycolysis. So if you're doing glycolysis, the cell must be in a need for ATP, because glycolysis produces ATP, among other things. So ATP concentrations tend to shut down glycolysis, as we talked about. AMP promotes glycolysis in the opposite way. ATP would shut it down. And in step 10 down here, remember, step 10, pyruvate kinase, is that feed-forward stimulation. If the product of step 3 builds up and step 10 is not keeping up, it will activate step 10, right, or enhance step 10. This was all a rehash of what we had in the last slide set. Let's look over at gluconeogenesis. The opposite should be true. If I have lots of ATP, do I want to do gluconeogenesis? Yes. I have energy right now. Let's not waste my pyruvate and try to make more. Let's store it as glucose for later. Right? So pyruvate converted to glucose, I'm willing to spend 6 ATP equivalents because I'm energy rich right now. So the molecules that indicate I'm energy rich, acetyl-CoA, which is the next, if you take pyruvate and do the next step, we'll talk about it on Tuesday, right, and turn it into acetyl-CoA, that's irreversible. So if I have enough of that, I don't want to turn pyruvate into acetyl-CoA, I want to convert it back to oxaloacetate. Also citrate, which is the next step in the citric acid cycle, the first step, generates citrate. So if you have a lot of that, let's shut down or promote gluconeogenesis and shut down glycolysis. Turn it into glucose. Go store it for later. I have enough for now. In the opposite scenario, if you have lots of AMP around, it means, hey, let's not waste this energy. We don't have a lot of it right now. So if you have lots of AMP, then you're energy deficient. If I'm energy deficient, I don't want to be wasting it on gluconeogenesis. I want to do glycolysis. So AMP shuts this step down, right? ADP as well as AMP shut down the opposites of step 10. We call it 10A and B, going backwards the other way. So all these make logical sense. If you want to make something, the pathway that makes it tends to get shut down by that product. The absence of that product, which is indicated by AMP here, tends to promote the pathway that makes ATP. Over here, lots of ATP says we should be doing this, so it's activated. We use lots of ATP to make this stuff. But the absence of ATP, in other words AMP and ADPs, shut this process down. We don't have the energy to do this. The materials that we make from the next step, we turn pyruvate into acetyl-CoA and into citrate, also promote this because I have enough. Don't turn more pyruvate into the stuff I already have. So all these should make sense, right? And all the regulated steps here are shown by the opposing arrows, the blue versus the orange, where the double blue arrows indicate everything that's reversible. What's here at the top is the only time we worry about this top step with the hexokinase and the glucose 6-phosphatase is in liver cells. We talked about that one. Okay? So some hormones that dictate to the cell what you should do besides this in gluconeogenesis, it's favored when we have lots of raw materials, lots of ATP, lots of biosynthetic precursors. Or if you eat a lot of insulin, right? So if you, or not eat a lot of insulin, you eat a lot of food and you release insulin because of the excess food, insulin tells the cell to ramp up the enzymes for glycolysis, for phosphofructokinase and pyruvate kinase. And the cell says, no, wait a minute, I don't want to do that. We have lots of ATP, right? Yeah, but you're about to get an influx of glucose. I'm just preparing you. That's what this insulin is saying. And you know how it works through the receptor. It releases the molecule on the surface that imports the glucose and the sodium together. So insulin also tells the enzyme, or tells the cell, to upregulate the number of phosphofructokinase 1s, PFKs, and pyruvate kinase, because you're about to need them, because a lot of glucose is about to enter the cell. Okay, so make sure you understand how those hormones work with this, how insulin works and how gluconeogenesis and glycolysis are reciprocally regulated. Okay. All right, let's look at blood glucose concentration, since we're talking about insulin and we're going to get to diabetes in a second. Let's see how this thing works. So one enzyme here is going to do two functions. Okay, this is not an enzyme we've talked about yet. This is a new enzyme. It's not in the pathways we just mentioned. It's kind of a helper to the pathway, right? So it's got two parts. It's called PFK2 and phosphofructokinase 2 and fructose 1,6-bisphosphatase. So let me explain what those mean. You know what PFK1 is. Phosphofructokinase 1. It's step 3 of glycolysis. It puts a phosphate on an already phosphorylated fructose. It's already got one on number 6. It's getting a new phosphate on number 1. PFK1 does that. This enzyme is called PFK2. It still uses the same substrate, fructose 6-phosphate, but in this case it puts the new phosphate on number 2 of the fructose, not number 1. Right, so the one that's now in the ring. So we make a fructose 2,6-bisphosphate. This is no longer a substrate for aldolase. It will not continue in the next step. Right, so what's the point of this? Well, let's think again. Let's say we have a ton of glucose just entered the cell. You had a dozen glazed donuts. Don't do that. Right? You had a dozen glazed donuts. What's going to enter the cell? A huge bolus of glucose. So all the hexokinases and glucokinases are working overtime to phosphorylate all that glucose. Now all of it gets turned into fructose 6-phosphate because our isomerase is favored to go in that direction by Le Chatelier's principle. Now we have all this fructose 6-phosphate that's being worked on by PFK1. And pyruvate kinase at step 10 is just not keeping up with it. So what happens is fructose 1,6-bisphosphate, the normal product, tells step 10, go faster. We covered all this already. Well, that's still not fast enough for a dozen glazed donuts, right? There's just so much glucose. So this enzyme, which normally gets nothing to do, much like glucokinase, sits around, lets PFK1 do all the work. It has a very high Km for fructose 6-phosphate. But in this case, there's so much of it around that PFK2 gets something to do. So it turns some of that fructose 6-phosphate into fructose 2,6-bisphosphate, which is no longer a substrate for aldolase. But what it does, it goes and binds to pyruvate kinase, step 10's enzyme again, just like the 1,6-bisphosphate did, but when the 2,6 binds, it's like supercharging it, right? It tells it you are really not keeping up. You should go a lot faster. So pyruvate kinase is normally going along at its regular speed. If the fructose 1,6-bisphosphate binds to it as an allosteric activator, it goes faster. But if the 2,6-bisphosphate binds, it goes really fast. So what this is kind of a safety net. Make sure step 10 can keep up with 3 by all means. That's what it's doing. Okay? However, we need to fix this problem at some point because we're making a molecule, fructose 2,6-bisphosphate, which is no longer useful as a metabolite. We can't digest it anymore. Right? It's good for telling step 10 to keep up, but we don't want that going forever. Once step 10 is caught up and we're really, as long as you don't eat another dozen glazed donuts, as long as you don't have another bolus of glucose coming in, we don't want to go that fast anymore. So we need to turn this fructose 2,6-bisphosphate back into just fructose 6-phosphate. We need to cut off this phosphate. Fortunately, we have an enzyme for that. It's called fructose 2,6-bisphosphatase. That makes sense. And in fact, it's the same enzyme. They're both parts of the same chain. So this enzyme has a two-phase functionality. One part of it is the kinase domain, shown in purple here, and one part, in the more pink color, is the phosphatase domain. It cuts off the very same phosphate that the kinase domain puts on. Now, you don't want both of these pieces working at the same time, obviously. That doesn't make any sense. It would waste ATP. So when the kinase domain is active, the phosphatase domain doesn't work. And when the kinase domain doesn't work, the phosphatase domain does work. So how do we know which one's working at any one time? Well, right here in the center between the two, you see the linkage. It's one long chain, so this thing doesn't have quaternary structure. It's one single chain, right? It does function as two of these whole things will come together and form a homodimer, but that's not so important to this part. But there's a serine residue right in the center here. And that serine is the target of a kinase. None of the kinase is seen here. It's just a kinase. We're not going to talk about which kinase. I don't want to confuse you. But it is a kinase. And that kinase puts a phosphate on the serine, or some phosphatase cuts it off. When the thing is phosphorylated, one of these two domains will be active and the other not active. And when it's not phosphorylated, the other scenario will happen. So we can control this enzyme. Right? They're on a single chain, and I either turn one on or one off. It's like a little switch in the center. Right? One's on while the other's off. If I am loaded in glucose, right, I'm trying to keep up, which of these two domains do I want active? The kinase domain or the phosphatase domain?
Kinase.
I want the kinase domain active, right? And if I have step 10 caught up now, and I want to slow it down so don't go faster than step 3, I want the phosphatase domain active. And that's all controlled by yet another pair of enzymes, a kinase and a phosphatase, that work on these guys. And those are again controlled by another set of kinases and phosphatases. And this gets very complex very fast. So we're not going to talk about all the behind the scenes stuff here. Just know that this one enzyme has dual functionality. It activates with the phosphate on it. It deactivates when I cut the phosphate off. It is a kinase and it is a phosphatase, but never at the same time. Okay, so this helps control how much glucose goes through the glycolytic pathway, and so I don't build up a lot of intermediates between steps 3 and 10. Okay. All right, one last topic about glucose, obviously, is diabetes. And so there's two types. Type 1, remind me, what's the problem in type 1 diabetics?
You can't produce insulin.
Right. For some reason, insulin is not being made, right? Whether it's because there's a genetic mutation in the sequence, or the insulin can't get out of the cells, in the islet cells. Whatever it is, there's no insulin, or a decrease in insulin in the blood. Okay? And so even though you eat a lot of glucose, then the cells are never told it's out there, right? So they don't import it very well. Their little sodium glucose importers don't get put on the surface. They don't get the signal. So there's plenty of glucose around, but the cells are not told to come get it. All right? What about type 2 diabetics?
Something wrong with the pathway?
Which pathway?
The insulin pathway?
Okay, what? Be more specific.
The insulin receptor pathway?
Okay, so the receptor could not work. That's true. What else? That's not the only possibility.
Anything else in that pathway, like any IRS molecules not functioning properly, then it'll shut the whole thing down.
Right, so this is going back to our cell signaling stuff. I don't mean by pathway, I wanted to be clear that it wasn't the glycolytic pathway or gluconeogenesis that we were just talking about. This is our cell reception pathway, right? The insulin receptor binding to the receptor tyrosine kinase and the autophosphorylating each other, and dimerizing and so forth. That signaling cascade is broken somehow, right? It could work just fine. And the body has been subjected to so much insulin over the course of its life that it becomes insensitive to it. That would also look like a type 2 diabetic. There could be nothing wrong with production of insulin, and there could be nothing wrong with the receptor pathway. It's just the body has become, it's like the boy who cried wolf. It's become so used to seeing, oh, the insulin says there's glucose out here. And the cell says, yeah, there's always that. I'm ignoring that. Right? So it becomes insensitive to it after a while. That's a common symptom when someone is on a very high glucose diet and becoming very obese, because you tend to store a lot of fats and there's lots of sugars around. So when you have the body type 2 either becoming insensitive to insulin, or in fact there could be a problem with the receptor, like you said. Okay. In type 1, it's very trivial to fix, but difficult to manage. So it's trivial to fix, you just add insulin, but you don't do it at any one time. You do it when the glucose concentration is spiking, so as you eat a meal, right? You don't want to do that when you're not high glucose. So managing it is more of a difficult thing, but the fix is rather simple. You just provide insulin. Type 2 is not so easy. Adding insulin does nothing because your body's already not responding to insulin, right? It's ringing the bell like glucose is out here, come get it, and the cells are ignoring it, right? They're not getting the glucose. So what does it look like inside the cell, right? So in the blood, there's tons of glucose. It's like syrup, right? But what does it look like inside the cell? There's very little glucose, right? It just trickles in. So you're always tired, right? That explains the fatigue on here. Can you explain the blurred vision? Why would you have blurred vision in type 2 diabetics?
Does it have to do with like the crystallization of glucose and like the cataracts? Right,
so sort of like our cataracts from the previous one, except it's not galactose this time, and galactose crystallizes rather easily. Glucose is very, very soluble, so you can get high concentrations before it forms crystals. But now your blood is very viscous, right? It won't flow very well. So in the very thin capillaries in the retina there, so it doesn't flow very well, the cells aren't perfused as well, and you're going to have a decrease in the function of the receptor cells for your vision. So you're going to get some blurry vision. Not so much that it's forming crystals, although it probably could if it gets high enough, but your cells are not flowing, the blood isn't flowing very well through those small capillaries. Why are you having excessive thirst? Why are you always thirsty? Let's see if we can explain that. Again, the problem here is your blood is full of sugar, full of glucose, hyperglycemic.
Would your blood have a hypertonic environment in relation to your cells, so it would like pull water out of the cells?
Right, so the blood is hypertonic compared to the interstitial or cells, and so since the sugar can't get in, the water is going to come out. So the water flows into the blood. So what's going to happen to the volume in the blood? It's going to go up. What happens to the blood pressure? It goes up. What happens to the interstitial fluid? It tends to become dehydrated because of that. And that's where your receptors are for thirst. So you're thirsty. So you drink some water, which explains the next step. Since you're drinking all this water and you don't need it in reality, your kidneys start filtering it out and get rid of it. So you get frequent urination. It's the same thing. Right? So let's go on to treatment. How do you fix this? Well, the first three are the thing you've heard forever: a healthy diet, proper weight, and exercise. Right? Those go without saying. That doesn't treat everything, but that's always included in every treatment, or almost every treatment. So you can treat some of it with drugs. And what we'd like to do is tell the cells to import the glucose. Well, that's great for type 1s. You just provide them insulin. You can't really give them the receptor that's broken. Now, there are some viral drug therapies that try to give the cells new copies of the gene that work, and that sort of works and sort of doesn't work sometimes. Or you could add drugs that work on the liver, which is what drugs like metformin do. So since your liver is doing gluconeogenesis, right, think about what's happening. The blood is full of sugar, but the cells, like muscle cells, are saying, hey, there's no sugar here, because they can't get it. They can't get it out of the blood. They don't know it's there. So all these bloods, all the muscle cells are releasing a hormone called glucagon, which goes to the liver and tells the liver, send us more glucose. Right? I mean, it's a reasonable response. There's no glucose in the cell. Shout for more. Although they don't know that there's plenty in the blood. So they shout for more, and the liver responds accordingly, releases more glucose, which is exacerbating the problem. So these drugs inhibit the enzymes of gluconeogenesis in the liver, right? They inhibit the production of PFKI and the glucose 6-phosphatase. Now, in a normal cell, doesn't make glucose 6-phosphatase, so that's not a problem. But normal cells will also make PFKI. In diabetics, however, that could be a problem, because if you inhibit that gene in cells that are not liver cells, now they have absolutely no source of glucose, and that could be detrimental. So you can't stay on metformin forever, right? It's not a forever treatment for diabetics. It is a short-term treatment, but because it inhibits PFKI in addition to glucose 6-phosphatase, you can't stay on it indefinitely. So hopefully it's a stopgap treatment until you get the top three working with a low glucose diet and weight loss and exercise and things like that, so that you don't need to stay on it as long. So type 2 diabetics, if caught early, are fully treatable, but not always. All right, so let's talk about some things that can go wrong other than diabetics. It's something called futile cycles. If I'm doing glycolysis and gluconeogenesis simultaneously, what will be accomplished? If you had to write out a reaction for glycolysis and gluconeogenesis happening at the same time, what's the only thing you'll accomplish?
Waste energy.
You'd waste ATP and GTP, right? So that's bad. Those are called futile cycles. They don't accomplish anything. All they're doing is hydrolyzing ATP molecules, okay? So this could be really bad, right? But hydrolyzing ATP isn't just hydrolyzing ATP. That energy has to go somewhere, right? It's an energetic release. It's negative 7.3 kilocalories per mole. And you're wasting 4 of those with every futile cycle. Remember, we get 2 out of it for glycolysis, but we spend 6 to make it back. So it's a net change of negative 4, right? We lose 4 each round. That energy goes somewhere, and it goes into heat, right? It heats up the cell. So we call this malignant hyperthermia, right? It's a increase in temperature that's malignant. It's bad for you, right? It shouldn't be happening. It's misaligned. It's however you want to say it, right? So these futile cycles are bad, so there's usually a problem, okay? Sometimes it's not a problem, right? So look at the last case, heat production. So we deliberately make this a futile hydrolysis of ATP to generate heat. When would you want to do this? When would you want to deliberately waste ATP just to make heat? It's an easy answer.
When you're cold?
Not necessarily when you're cold, but when someone is cold. Right? When you're cold, we can be much more efficient at the wastage of ATP by shivering. Right? We funnel that ATP into muscle contractions, which are much more efficient use of the energy. But just hydrolyzing an ATP to generate heat isn't as useful. So who would need it when they're cold? Not you, because you can shiver.
A baby, maybe?
How old is this baby?
Still young enough to breastfeed, or maybe in the mother?
Well,
Still in the womb?
if it's still a fetus, then it's in a 37-degree oven. You're good. You're not going to get cold. Right? But let's say you just came out of the oven, right? You were a newborn. You were at 37 degrees, happy as can be, and now you're at room temperature, right? Instantly. Well, that's chilly, right? You've been at 37. You've been not having to regulate your internal temperature so much. So you need to find a way to warm up, and you have absolutely no control of your muscles yet. Right? If you've ever seen ever delivered a newborn, nothing works. No limbs are under its control yet. And so they have to learn those muscle controls. So here's one way to generate heat. We'll talk about another one later. But heat production is the deliberate hydrolysis of ATP to generate heat. If you're doing that as a 20-year-old adult, that could be a bad sign, because you can shiver much more efficiently than doing this. So if this is happening, that's malignant hyperthermia. Right.
Somebody who's paralyzed do that or no?
Someone who's paralyzed probably would try to shiver still, because the muscles may not work or the nerve connection may not work, and it's unlikely they'll go through the malignant hypothermia route because you're not supposed to do that. Right. So lastly, let's talk about lactate. We did this on the other lecture, but I'm going to round out with this. One of our fates of pyruvate was to form lactate, right? So we take pyruvate and reduce it to the alcohol lactate instead. And it's a dead end of metabolism because there's nothing you can do to it, right? There's no carbonyl anymore. There's no place to attack it. It's just a dead end molecule. So what we do is we send all that lactate through the blood back to the liver, and lactic acid or lactate is a buffer, right? It's got a PKA, it's a carboxylic acid, and so it's going to drop the pH of the blood if there's lots of lactate in the blood. So this is after a strenuous exercise. You will make lots of lactate because the muscle needs to work now, but all that lactate will cause your blood pH to become slightly more acidic. You'll buffer it with bicarbonate, but all that lactate eventually makes its way to the liver, and after your half marathon you just ran, right, you're tired. You should not just stop, but maybe walk a while to get all this lactate out of the muscles and through the blood to the liver. You don't want cramps. And then finally, the liver is going to convert all the lactate back to pyruvate, back to glucose, at the expense of 3 times the amount you're going to get out of this glucose later. So after the marathon is over, right, you thought you spent a lot of energy then. So the last thing you want to do after running this half marathon is go out drinking, because your liver has got plenty of work to do already, right? It's working harder than it did during the exercise. So now if you add a lot of ethanol to it and have it work on detoxifying that at the same time, your liver cells are going to die, right? You'll eventually get cirrhosis of the liver. So after exercise, treat your liver nicely, because this Cori cycle is generating the glucose back to the muscles at 300% interest from the liver. Okay. So if here's another graphic showing the same thing. I just want to show it to you a different way. So if you have a cell that's producing lactate, those are going to be mainly muscle cells and exercise, and red blood cells and brain cells and so forth. They export it into the bloodstream. It goes to the liver, right? The liver imports that lactate and turns it into all the things that you might need, including glucose and other amino acids, and then sends the glucose out in the blood to cells who need glucose again. So the liver is constantly monitoring this to make sure that you need to either eject glucose into the blood, take up glucose from the blood, tell the cells that, hey, you need to send me your lactate, and so forth. So glycolysis and gluconeogenesis aren't on-off switches. It's not like we're only doing one and not the other. They're both on all the time. It's just a matter of how much on are they. We want to turn one down, but not off, and turn the other up. But they share 7 of the 10 reactions in common. So we're just shifting the equilibrium one way or another, not by adding enzymes, but by changing the number of substrates and products on each end, the glucose and the pyruvate and the lactate and so forth. So we are altering the equilibrium, but not through adding enzymes, but by altering the concentrations of all the molecules. Okay? So the fact this is going on both ways at the same time prevents any lag period. So if suddenly you're doing glycolysis and you need to convert to gluconeogenesis, there isn't this huge lag of having to do 10 reactions before we see a product. Everything just shifts the other direction because there's a lot of intermediate molecules built up there. And why is this a good thing? What organs and cells are happy that you can shift quickly? Well, things like the brain and red blood cells, because they need a continuous supply of glucose. If you have a lapse in it, if it's brain cells, you're going to pass out. Okay, we need a continuous supply of glucose.